Hello and welcome to the C22 neural signaling video.
Strong start.
Standard 1. Neurons. What is a neuron? That's a great question. Neurons are nerve cells that carry electrical impulses. So they're cells in the nervous system. Oh no, I spelled system wrong. Nervous system that carry electrical impulses. So an electrical impulse is like a difference in voltage. That's going to be really important as we go throughout this unit. And what you need to know about neurons is you need to know their relative structure. So they have, starting from left to right, we have dendrites, a cell body, and then an axon. So these are the three parts. So we always like to start with the cell body. It's the easiest, and then the axon. So it looks something like this. In the cell body, you have a nucleus, right? You'll have a lot of mitochondria. Okay. And then your dendrites, like, stick off, they branch out and connect neurons to other neurons. So you get all of these little tubes that look like this. And so you got your dendrites, your cell body, and your axon. The end of the axon is often called the terminal button. We'll see that later on. And so what happens is the electrical impulse will move along an axon from dendrites, along a neuron from dendrites to the axon. Now, how do we actually make this happen? Well, it all starts at resting potential. Resting potential is the state your axon is in most of the time. So we're going to zoom in on the axon. And so inside your axon, what you'll find is that you have these pumps. So you have these pumps all along the axon, and these pumps are responsible for maintaining the voltage difference between the inside and the outside. So whenever we talk about the voltage, we always talk about the inside. And so for resting potential, the inside has negative 70 millivolts. So it is more negative inside than outside. So that means there's negative molecules inside the axon, like DNA is negative, that helps make it negative 70, and there's more positive charges outside than inside. Well, why are there more positives outside than inside? Well, they should be the same because of diffusion, but what you're doing is you're using protein pumps. And you're using a specific one called the sodium-potassium pump. The sodium-potassium pump will send 3 sodium out for every 2 potassium it brings in. And so you're going to have potassiums inside here, right? And sodium outside here. More sodiums outside than potassiums inside because you're doing it in a 3:2 ratio. So the more you do it, you get a lot of sodium out here, creating that positive charge accumulation outside and less positives inside for about negative 70 millivolts. We call this membrane polarization, and so resting potential, we say the membrane is polarized. And so for our class, polarized means it's negative, and it's roughly negative 70 millivolts. And so that is because of the difference in positive charges and the permanent negative molecules inside of the axon and inside the cell. Now, we start the action potential. So what happens next is, standard 3 is all about a nerve impulse. And so a nerve impulse is when action potential move along an axon. And so what happens is you have your axon and you let in positive charges and you're letting them in, and then that'll happen down the axon and it'll keep happening. And so this is called propagation. So this is called propagation. And so it'll jump along the axon. And so what you're doing is you're sending the signal that way. So a nerve impulse is when the action potentials move along an axon through the nerve fiber. Now, there are different things that can affect the speed, okay? There are different things that can affect the speed. The first one we'll talk about is myelinated versus non-myelinated, or unmyelinated. The one above here is unmyelinated. It just keeps happening along it. In a myelinated neuron, you have myelin covering it, and it can only happen at the nodes. So instead of happening along every single part of the axon, it only happens at what are called the nodes of Ranvier. And so it'll enter here and here and here. And so it's jumping. So instead of it moving along it in a smooth motion, the signal is jumping along the axon to these spots here. And so that will speed it up and cause it to take less energy, which we'll see more about later. Now, other things that affect it are size of an animal. So for size of an animal, it's correlated like this. Speed will decrease as the animal gets larger. And the last one is the axon diameter. And so for axon diameter, it's the opposite. So as the diameter gets larger, the speed goes up. Okay, so these are unrelated. So this is diameter of an axon, and the other one is speed of the animal. You can see how they have different relationships. Okay, so now that's basically what you need to know for SL, for moving it along an axon. Now, for between neurons, we have what are called synapses. So a synapse looks something like this, right? You have your axon, you have the end of an axon, and then you have the dendrite of another one. And you have this gap here is called a synapse, sometimes called the synaptic gap or the synaptic cleft. It goes by like 3 different names. Fun stuff. And so now this is a junction between the cells. So it's a junction between the two cells. There is a gap between them. They don't actually touch. And so what happens is in most synapses, the signal will pass from one to the other. So we call it the presynaptic and the postsynaptic. And then what happens is you send a neurotransmitter across the gap. And so the neurotransmitter will go from one to the other. And so now 6 and 7 we're going to do together. We're going to do those two standards together. And we're going to zoom in so that we can really see what's happening here. So standard 6 is basically the first half of this, and standard 7 is the second half of this process. So first what we have is we have the action potential moving down the axon. So it moves to the end. When the action potential reaches the end, it causes a positive charge on the inside of the membrane. That positive charge causes a calcium channel to open. So the action potential reaches the end, that's step 1. Then a calcium channel will open, that's step 2. And calcium will go in. Calcium in. So you can have calcium come into the cell here. Now, the third step, that calcium will react with a vesicle, with a neurotransmitter. So it'll react to the vesicle and trigger vesicle release. So calcium will trigger the vesicle release. And then step 4 is exocytosis of the neurotransmitter. And so it'll come, it'll be like here, and it'll spit it out into the gap. Then your neurotransmitter will diffuse across the gap. Oh wait, hang on. That is the end right there. So step 4, that is the end of standard 6. Now, starting on standard 7, it'll diffuse across the gap, or cleft. Okay, it'll diffuse across the cleft, and it will bind on a receptor. And so it will bind to the receptor here, and when it binds, it will trigger, so 6 is bind to the receptor, and then step 7 is trigger a new action potential. And it does that by opening sodium channels on the postsynaptic neuron. I think one of the really, really important things, right, the receptor is also on the postsynaptic neuron. IB will ding you if you don't say postsynaptic or presynaptic. So you need to make it very clear you understand which neurons you're referring to. Are you referring to the presynaptic neuron or the postsynaptic neuron? And if you don't do that correctly, you'll lose all the points. Okay, let's see. There are many types of neurotransmitters. The one that I have drawn as our example is acetylcholine. Acetylcholine. So acetylcholine is just kind of a basic neurotransmitter. It just causes an action potential. If you're in HL, we'll talk about it more in a second, but if you're in HL, you should already know what else it means to be that one. All right, so that is the end of the SL standards. So now we're moving on to the HL standards. So for the HL standards, we're going to start with number 8, which is all about, well, wait, we said there's an action potential, but we didn't really talk about how that action potential happens. So you start at resting potential, which we already talked about. From resting potential, you go to depolarization. Depolarization is really the first step. So resting potential is kind of like step 0. Depolarization is really the first step of the action potential. So this is step 1. And in depolarization, you have your, something causes your voltage to approach what we call threshold potential, which is around negative 50 millivolts. We call that threshold potential. When it reaches, when the inside of the membrane reaches negative 50 millivolts, this causes the sodium ion channels to open because they are voltage gated. So they open up and they let sodium in. This is going to cause the membrane to become depolarized or positively charged. So let sodium in, membrane inside becomes about positive 30 millivolts. And so you went from negative to positive, hence depolarization. And remember, this is a voltage gated channel, so it only opens because you've reached that threshold potential. Then when you get to the top, we have the next phase, which is repolarization. Repolarization is all about, okay, we sent the signal, now we need to make it negative again. Well, this happens when you get to positive 30 millivolts. That triggers the opening of potassium channels. And these are going to let potassium out. So both of these are going with the gradient. If you remember from resting potential, we created this situation where there was lots of potassium inside and lots of sodium outside. So when you open these channels, they go in the direction of the gradient. So in this case, potassium is going out, and that's going to cause it to become very negative. So the membrane is going to go negative, and most of the time, it actually is going to go past negative 70 down to negative 90-ish, and we call this hyperpolarization. Hyperpolarization is a method of preventing reoccurring action potentials, and so you don't keep sending it over and over again. So that is standard 8. So it's all about the potassium and sodium and which way they're going. Standard 9 is, well, how does it move along the axon? Well, what happens is, propagation is the word we already saw. When sodium enters, it's not just going to stay in one spot. It's going to slowly diffuse down the axon. And as it diffuses down the axon, it will trigger the opening of more sodium ion channels. Because you're putting positive charges over here, that's going to raise you to threshold potential, and it's going to cause propagation. Okay? Now, I'm going to skip to standard 11, because standard 11 is like, well, wait, what if you have myelination? And so if you have myelination, we talked about this a little bit already, but we have what are called the nodes of Ranvier. And these are the only spots where action potentials can happen. And so now the signal will jump like this, and so you have all of your channels and everything right here, and your pumps, and so you have limited where it happens. And this is called saltatory conduction. Saltatory conduction. So this, instead of it just propagating along, it's now jumping. And this is going to save energy and make it faster. So it's got double benefits. So a large amount of your neurons are myelinated because it makes them save energy and they're faster. A lot of neuro disorders are actually the breakdown of myelin because then they don't work the way they're supposed to. Alright, number 10. I really like standard 10 because if you can do standard 10, it shows that you understand action potentials. And so there are these things called oscilloscopes. And oscilloscope traces are what you get when you hook it up and you measure it. And so it looks like this. And so you should be able to label the different parts. And so we have here, right, so this is resting potential. So this would be at -70. Here's our threshold. Here's our top. Okay, so this is resting potential. Then that means that this part here is depolarization. And then on the other side, we have repolarization. And so you should know what's happening at each. We just talked about it, right? So depolarization is going to be our sodium going in, and our repolarization is potassium going out. And that's going to give us that pattern. That is our oscilloscope. Okay. Alright, the next three standards, 12, 13, and 14, we're jumping back to our synaptic transmission. And so we're going to start with, we'll start with 12. So 12 is our standard on exogenous chemicals. And so these are chemicals that you put in your body or in an insect's body that have some sort of effect on it. And there are two different ways. And so they can either block synaptic transmission. That's one category. Or they can block reuptake of a neurotransmitter. And both of these are going to have different effects on your action potential and your ability to send signals between synapses. And so if you can't send signals between synapses, you can't pass that message on to the next neuron. And so for the first one here, when you block synaptic transmission, what you have in this specific example is an inhibitor binds to a receptor on the postsynaptic membrane. So normally you bind the neurotransmitter there, but instead you're binding an inhibitor. This then prevents signal, and it can lead to paralysis and death. Our specific example for this one is our neonicotinoid. I hate the spelling of this. Neonicotinoid. There we go. Insecticide. So these are for insects, and this is one of the ways we use to kill insects. The reason it leads to paralysis and death is because you cannot break down the inhibitor. Cannot break down inhibitor. Now for your normal neurotransmitters, you break them down and then you send them back to be reuptaked on the other side. So acetylcholine is broken apart into acetate and choline, and then you send it back to the other side. This allows you to stop the signal as well as send it back to the other side so it diffuses against the gradient. But in this one you cannot break it down because it's not acetylcholine, it's neonicotinoids. So this one binds to your acetylcholine receptors. So this one is specifically blocking acetylcholine, which can be abbreviated as ACH. Okay, so it's blocking ACH. The other one, the other type of exogenous chemical, blocks the reuptake of the neurotransmitter. And so what's happening here is you are leaving the neurotransmitter. You're leaving the neurotransmitter in the synapse. And so instead of you taking it back to the presynaptic neuron, it's now staying in the synapse. It's staying in the synapse, which means it's going to continue to trigger the postsynaptic neuron. So the previous one stops the signal. This one is overstimulating the next one. And so it's sending that impulse over and over again on the other one. And our example for this one is cocaine. Cocaine is not permanent like the other one, it's temporary. And so what it does is it makes the dopamine stay in the synapse. So it blocks dopamine reuptake. So dopamine reuptake is stopped by cocaine. It also causes it to secrete all the dopamine it's got, so it floods that synapse with dopamine, and it makes you really, really happy. But this leads to addiction, and it causes you to have a hard time feeling a natural high. And so it has a really negative effect in the long term especially. So those are the two types of chemicals that can affect synaptic transmission. Now, for the next one, 13 and 14 kind of go together, so we're going to do both of them. There's two main categories of neurotransmitters. There's excitatory. These are acetylcholine is an excitatory one. Oops. And there are inhibitory neurotransmitters. Oops. And inhibitory neurotransmitters. So these are going between the synapses, etc. So this one is acetylcholine, and our example for our inhibitory one is going to be GABA. And so these ones are trying to cause an action potential. And so they're trying to make it more positive. So they want to make it positive. GABA, on the other hand, is trying to suppress an action potential. And so they are going to make it more negative. And so what you do is you sum them up and you see which one is greater, and then that'll tell you whether it's going to go or not. So you sum the positives and the negative effects of the acetylcholine and the GABA, because they can both be released at the same time and they can both be having an effect. And so that's standard 14 is you take it and you go, okay, and it leads to an all or nothing. So if you reach the threshold, it will go. If you don't reach the threshold, the signal will not be sent forward. All right, standard 15. Standard 15 is all about pain, and it's funny, we actually talked about it in C3.1 a little bit too. It's all about free nerve endings. And so free nerve endings are just the ends of neurons that are in your skin. At least for this one, we're talking about in our skin, and they can sense certain things. So they have receptors. They have receptors that are triggered at certain things. So those could be temperature, acid, capsaicin, which is in chilies, acid, and like burning, right? So excess heat as well. So they have these receptors, and when the receptors, they sense it, they open positive ion channels, and they flood it with positive ions. Well, if you're letting in positive ions, and it could be like any positive ion, you let these in, this is going to cause an action potential, and then that action potential will be propagated and it will eventually reach the brain. And so remember, if you think back to the other one, the white matter takes the signal to the brain, the gray matter does the reflex arc. And so they're all triggered by the free nerve endings, but they go to different directions. So last time we learned about the reflex arc, today we're saying, oh, well, it triggers an action potential that eventually makes it to the brain. And so that's how we feel pain, is because of these free nerve endings in your skin. It's kind of crazy that it's that simple, but really complicated. Speaking of simple but really complicated, we have consciousness. And so This is another example of an emergent property. So you take a bunch of neurons, you stick them together, and you somehow get consciousness. And what we talked about before is the interaction between the different neurons in just the right way allows us to be able to think and to be aware of our thoughts and that kind of a thing. And so this is a really good example of another emergent property. So you take a bunch of neurons, you stick them together, and you get a brain. And then because of the way the connections are formed, those connections allow for something that is greater than the sum of the parts, just like what we talked about with our bodies working as organisms, and like our simple example of like taking apart your phone or whatever it is. So it has to be put together just right for it to work, and then we get consciousness. And that is the end of C22.